So transposable elements are the elements which can transpose, that is they can move from one place to another in the genome, and then they can cause the repetitive elements or the repetitive DNA within that genome. Simplest example of them is insertion sequences which are related with the prokaryotes. They are small, their sizes they range from less than a kb to maximum is 2 kb, and they are flanked by invert repeats, and they encode at least one gene that is responsible for their transposition. Generally they don't encode for other functions or additional characters. Here we see the structure of a insertion sequence, IS elements. We have those invert repeats which are anti-parallel or facing each other, so repetitive elements. These are the distinctions in them. We have a transposase gene, so that gives the transposition properties to this transposable element. And then there another distinction is when they get into the host genome, they cause target site duplication. So normally they create the sticky ends in the genomes of the host, and then later on when the complementary nucleotides are formed, they become those duplications. Transposons are relatively complex as compared to the simple IS sequences, and then they encode for additional characters in addition to the genes responsible for their transposition. Here in this diagram we can see there are two major classes, class 1 and class 2. We have within class 1 we have long terminal repeat retrotransposons. So retrotransposons are the ones that have the ability of reverse transcription. So they have RNAs, and then those RNAs they get converted into cDNAs, and then in this way they replicate. So they have long terminal repeats in their end. So we have those long terminal repeats, one is here, one is here. So they are not invert, but they are kind of forward repeats, they are in the same direction. Then we have in the another category, we have non-LTR retrotransposons which do not have those long terminal repeats. So the examples are lines, which are larger ones, and sines, which are comparatively simple. So line stands for long interspersed nuclear elements, and sines stands for short interspersed nuclear elements. So in addition to that, we also have DNA transposons. Eukaryotic transposable elements, as we have seen earlier, they have two classes. Class 1 is they have a reverse transcriptase in them, so they have RNA mediated mechanism of transcription. Class 1 can be categorized into LTRs, long terminal repeats retrotransposons, and simply transposons. And then we can also have retrovirus-like elements. Sines and lines, as we have seen earlier, they are present in the non-LTR retrotransposon category. The sines are 80 to 300 base pairs. One example from them is ALU repeats, which are present in the humans, and then these lines are 6 to 8 kb. They are relatively longer. The examples of class 2 transposons are ACDS, which were studied in maize. Actually, the work of Barbara McClintock that led to the discovery of these transposons, in which you can see the role of ACDS elements in maize. They give different characteristics and traits to the kernels in the maize. The other example is P elements in Drosophila. You can look into the literature for them. Then there are additional elements, we call them as mites, which are miniature invert repeats that have the features for both class 1 and class 2, and they are 400 base pair long. Here in this diagram we see the comparison of human, yeast, and maize and E. coli. And in the blue we see those repetitive regions, the transposons. So we see large number of blues in humans. Reds are exons, whereas the oranges are introns, and these are the pseudogenes. So we see less proportion of those transposable elements in yeast. Let's look into the maize, the huge one, the big proportion. So we are here comparing actually the 50 kb region of the genome, and again in E. coli they are less, and we see IS elements in E. coli. So transposable elements, they make up the significant contribution into the genome of the organisms, specifically in the eukaryotic organisms, and then they move across the genomes, and then they cause genome expansions.